Thank you, everybody, for joining today's webinar called Using eDNA for Wind Energy and Wildlife Studies. I just want to give a quick introduction on WREN and then introduce our speakers, and then we'll have them present. Let's see if I can advance slides. Okay, so WREN is designated as Task 34 of the IEA Wind, or International Energy Agency Wind Technology Collaboration Program. And it began in 2012. And since that time, WREN has published 4 papers, 4 technical reports through the International Energy Agency, 4 fact sheets, 12 research briefs, has hosted 24 webinars, 3 expert forums, and is the host of the technology tool, which can be found on the TETHIS website. The work that WREN has focused on includes both kind of broad topics like cumulative effects, adaptive management, the mitigation hierarchy, but also species-specific interactions with wind energy, or species group interactions like bats, marine mammals, birds. And so the work also is split between land-based and offshore wind energy interactions with the environment. And so the current task 34, recently ended, and we applied for a new environmental task within the IEA Wind Technology Collaboration Program, and were approved. And so we're now Task 59. We're keeping the same acronym, WREN, but we are changing what the acronym stands for. So originally, WREN stood for Working Together to Resolve Environmental Effects of Wind Energy. We switched that to Wind Energy Environmental Research and Engagement Network. And this new task for WREN was approved for 4 years. It just began at the 1st of October. And the rationale for continuing this work is that wind energy and environmental interactions persist, and that international collaboration is necessary to advance the solutions that we need. Because wind energy is global, and because habitats, ecosystem processes, wildlife cross boundaries, we need a broad set of expertise to advance these solutions. Going forward, for Task 59, we do want to carry over some of the work that we were doing with Task 34, so there will be some similarities, some consistencies. We will continue the TETHIS website, which is managed by the Pacific Northwest National Laboratory. We will still produce state of the science projects, have webinars, disseminate research brief publications, and so on. We are going to, in part, have a focus on the findings of the Grand Challenges Revisited report, which came out last year, November. The report is called Wind Energy Research Needs for a Global Energy Transition. This was derived from a meeting held in March of 2023, where researchers from 5 different areas came together to not only address the grand challenges within their group, but also to work together to find out where we can have interdisciplinary research to meet our renewable energy and our wind energy goals, but also to advance the state of the science for each of these categories. So there was an environmental group, social sciences group, researchers focused on the plant and grid, on wind turbine design, and on atmospheric sciences. And one other thing that we're hoping to work on going forward is to increase our engagement with countries that are not part of IEA Wind. Currently, there's only 22 countries participating in the IEA Wind TCP, but so that leaves a lot of the world that we can interact with and exchange information and lessons learned from the research that IEA Wind is doing, but in exchange, hear what other countries are doing and what they're learning. So that's just a brief overview of what we have going forward with WREN. I'd now like to introduce our speakers. We have two speakers today to talk about using eDNA for wind energy and wildlife research. The first will be Jason Adolf, professor of marine science at Monmouth University in New Jersey. He's been there since 2017. And before that, he was a faculty member of the University of Hawaii Hilo Marine Science Department for 9 years. He is a broadly trained biological oceanographer with a background in phytoplankton ecology, harmful algal blooms, and using eDNA metabarcoding as a non-extractive means of assessing marine fish community composition. Dr. Adolf currently leads Monmouth University's efforts to integrate eDNA as an integral part of fisheries monitoring plans for offshore wind development on the mid-Atlantic continental shelf, collaborating broadly with academic, state, federal, and industry partners. Our second speaker will be Dr. David Mouillot, professor at the University of Montpellier in France. He is recognized as a highly cited researcher in ecology and environment since 2016, with more than 350 international articles published and 28,000 citations. More recently, he has led the development of a new generation of tools based on environmental DNA, or eDNA, released by all organisms to monitor marine biodiversity across habitats and depth gradients. He highlighted an unseen dark diversity that divers and cameras miss. He also developed new models and artificial intelligence towards a better prediction of marine biodiversity based on a seascape approach and satellite imagery. He now holds a prestigious European Research Council grant. So with that, Jason, I will turn it over to you.
Thank you, Chris, and thanks to WREN for hosting this event. I'm going to share my screen. And Chris, can you confirm that you're looking at my title slide?
Yes, we see that and we can hear you, so go ahead.
Okay, great. Thank you, everybody. Thanks for coming. I'm Jason Adolf. I want to acknowledge my co-authors here. Keith and Shannon are co-PIs on this project. And Erin, Sam, Kieran, Nick, and Emma are absolutely integral to the stuff that we do. And it's my privilege to present the work that everybody has contributed to here. So offshore wind development is happening in response to climate change and the climate crisis. It's planned widespread on the east and west coasts of the US. There are a few actual turbines installed here, but each of these polygons are a planned wind energy area. We're working mainly in this area here off the New Jersey coast, the continental shelf. And with this planned response to the climate crisis, it's a fair question to ask, will the response have any kind of impact in itself on other aspects of the marine ecosystem? And we asked that in what are called fisheries monitoring plans, which basically are asking what fish are there and what effects might wind farms have on them. These effects have been looked at and considered by many people. Many of them are listed here in terms of direct effects. But we also know there are potential indirect effects where oceanographic conditions may be changed due to the presence of turbines and indirectly affect fish and other marine organisms. And we also know that the studies that we're doing have to be designed so that we can detect potential changes in wind farms against a background of high natural variability, global climate change effects that both have been well described in this area. So obviously, these are very complex questions and require a lot of data. And a lot of places where we don't have a lot of data, and eDNA is here to help. A sentiment that's shared by our countries. The U.S. recently published a national aquatic eDNA strategy that highlighted renewable energies and offshore wind development as a use case for this technology. You can grab that if you haven't read it at that QR code. We'll link you to the National Aquatic eDNA Strategy published in June of this year. So background on eDNA. eDNA is DNA that's collected apart from the organism that produced it. In terms of fish, it comes from sloughing off of mucus, body fluids, tissue into the environment. It has a half-life in the marine environment of about 1 day, which is a good thing because we know we detect it. It's been there recently. We collect this by filtering water from aquatic ecosystems and extracting the DNA into an environmental DNA sample. Most of the sampling that we do is bottom waters, because a lot of the work that we do, as you'll see, is trying to understand how eDNA compares to trawl surveys. The analytical workflows from eDNA, starting with isolated eDNA, we can think of it in two paths: qPCR with specific probes for specific taxa or small groups of taxa, and then metabarcoding, which is when you use more universal or degenerate primers and amplify and then sequence the diagnostic genes from a group of organisms such as vertebrates or fish. Today I'm going to be talking about what we've been doing with metabarcoding in this context, mostly. Each of these techniques leads to different sorts of analyses, as you can imagine, more presence-based analyses for either technique. But I think one of the strengths of metabarcoding is a community-based analysis using multivariate indices, which I will talk about towards the end of this talk. How we do this matters. I'm not going to go into detail on this, but for those of you out there who are interested, and it matters not just for the QA/QC of the data itself, but also for the intercomparability of different people using metabarcoding in different areas. So I just want to mention that. Our group here at Monmouth has been doing eDNA on the shelf since about 2019 in the context of offshore wind, and you can see all the places that we've sampled, including the bay. That's a bad coordinate with in the Empire Wind area with Inspire Environmental out here. This is Ørsted Ocean Wind 1 back in 2021. And eDNA is a great method, but it still requires physical collection of samples. And that's a challenge when we're aiming to cover this entire shelf area where all these wind area polygons are located. We're using small boats to do that now, which helps. We're using community and commercial partner scientists, and we're pairing with ongoing state and federal surveys to get as many samples as possible. But we see, and Dr. Mouillot is going to talk about, what we really need to start doing, and we're planning to do, is using AUVs to collect eDNA in to cover the space and time that we need covered. So I'm going to highlight a project that we're doing with the New Jersey Resource Monitoring Initiative, or RMI program, where we're sampling eDNA in 7 different campaigns. The blue dots on this map surrounding the Atlantic shores and ocean wind Ørsted areas is our offshore wind development area, and then we sample along the coast as well in the surf zone, because it's important that eDNA is one technique that can be used both offshore and in the surf zone to get comparable results. We also had community scientists, some of whose names are shown here in names. They're also co-sampling the surf zone with us, piloting a program to get community scientists able to produce eDNA data. Then we pair with 3 different ongoing New Jersey surveys: ocean trawl, which takes place between 0 and 30 meters depth along the coast, the entire coast, the Raritan Inventory Project up here in Raritan Bay on the New Jersey side, and the artificial reef surveys, which take place at artificial reefs off of New Jersey here, where we're pairing eDNA sampling with their ongoing capture surveys. And then my partner, Keith Dutton, does a lot of acoustic telemetry in the same area. So we're also pairing eDNA with those results. And I put this up here to mention that we collect environmental parameters, which are important in letting us deconvolute environmental versus wind farm effects down the road when we finish our analyses after the wind farms are built. So I'm going to show you some data here comparing from the New Jersey ocean trawl, metabarcoding eDNA results versus the trawl results. And these are paired samples. And the pattern you're seeing here in 5 different sampling bouts. We don't have the Bank data yet for April, so it looks like they're the same, but is that we generally see on a per sample basis more taxonomic richness in an eDNA sample than in a trawl sample. And that's a consistent trend that we've seen here and in other work that we've done. If you look at cumulative richness, considering n number of samples on the x-axis here, you'll see that trawl richness maxes out, and I showed that with a dashed orange line. But the cumulative richness in the eDNA samples surpasses that level much sooner than the number of samples required for the trawl, and ends up at a higher number. So we can replicate the trawl richness in terms of biodiversity with fewer samples, and with the same number of samples, we get a higher cumulative richness out of the samples. Now, differences in richness imply differences in things being caught in the eDNA versus the trawl. So these plots are showing on a sample by sample basis. This list here are the taxa that we most often find in the eDNA without a match in the trawl. And you can see the proportion of samples in which this happens is relatively high, up to 50, 60% for some taxa. And it makes sense that things like bay anchovy or strong swimmers like sturgeon or structure-oriented organisms like tautog would be detected in the eDNA but not in the trawl. On the other hand, in the eDNA, there's some things that come up in the trawl that we don't see in the eDNA on a sample-to-sample comparison. The bars in red is a simple answer. We don't have reference sequences for those organisms, but they catch them in the trawl. So we're never going to see them until we get a reference sequence for them. But there are other things that are less easy to explain, like the winter skate or little skate ASV that they catch in the trawl and we don't detect. But for the most part, the proportion of samples in which this happens is very low compared to the converse. We tend to detect most of the things found in the trawl samples most of the time. And then while metabarcoding is not quite there yet in terms of absolute abundance metrics, in terms of relative abundance within a sample, we do find in two different trawl eDNA comparisons. This was the Ocean Wind 1 that Rutgers did the trawling for us on. If you look at the relative abundance of taxa, you do find a regression, a relationship between that and the relative abundance of DNA reads in the eDNA metabarcoding data for two different surveys here. So to wrap this part up, we're very interested in comparing capture standard techniques with eDNA metabarcoding, and we're finding that the concordance between the two is generally good. We have other data. Something I'm really interested in is, of course, what don't you find in the trawl that you find in eDNA, which I'm not presenting here today, but happy to talk about. Okay, and then I mentioned community analyses. And I think that's a real strength of metabarcoding, building pipelines that go straight from metabarcoding data to multivariate or machine learning algorithms that tell you about community composition. If you're familiar with community composition, it can be quite confusing to look at. This is a three-dimensional plot of ocean trawl communities, and the color codes are the different sampling bouts, and they kind of cluster out, but really took 5 dimensions, which I can't show you to suss that out. What I am going to focus on is how we're using k-means clustering to label community composition from within a sample so that we can visualize it more easily. So what we're looking at here are 4 sampling season maps, and everywhere you see a different color on the map is a different community composition as determined by k-means clustering analysis of eDNA metabarcoding data for that station. And you can see pretty clearly some inshore-offshore differences here as well, clusters of organisms, a different community composition. The heat map on the right here is where you look for the cluster and it tells you which taxa are more abundant as a relative abundance within that cluster. I want to highlight the fall 2023 survey. This is the first time we got data back from the New Jersey Artificial Reef Survey, shown in these 2 shape files, a little red thing here and here. And what we found was that on that scale, in sampling the water column, we found a very different community composition in the eDNA that was dominated by things like eel, black sea bass, and tautog, structure-associated fish, that really told me, for one of the first time, something about the real life spatial resolution that we could get out of eDNA, being able to resolve those fish over a reef as compared to the surrounding environment. Another look at the artificial reef survey. Of course, in their ventless traps, the richness, few things pot in their surveys, and they're shown here, but the eDNA reveals a much richer community composition, including this taxon cunner here that their divers were telling me they see all the time, but they don't catch in the pots, and it was satisfying to find them in the eDNA. So to wrap up with my AI generated of what is metabarcoding of fish image here, we're using eDNA as part of fisheries monitoring plans. We're very interested in establishing ecological and methodological baselines for the methods. We're focused on understanding the gear bias differences between eDNA and capture surveys so that we can better integrate the two together. And we're thinking about what are the best ways that we can implement eDNA in fisheries monitoring plans, like community monitoring and sampling from AUVs. Alpha and beta biodiversity are great strengths of metabarcoding, and so is community science that I didn't get to talk about today. But I'm happy to answer questions about. Another strength of eDNA is that you catch things you detect things in the eDNA, such as bottlenose dolphin, which are very abundant in terms of genes. And if you go out on a boat ever in the mid-Atlantic, you know they're very abundant in the ecosystem that you don't want to catch in your trawl, but they're an important part of the ecosystem. So there's all these things like humpback whale, fin whale that are important that you pick up in eDNA, but wouldn't necessarily pick up in a trawl or capture survey, thankfully, sometimes. So the great thing about eDNA as a fisheries monitoring tool include its resilience, it's easy to collect data, and it's easy to access impacted areas such as shorelines and built wind farms. I believe Dr. Mouillot is going to talk about that next. It's scalable. It can be collected from AUVs. This is the MBARI SOB AUV that we're trying to partner with in the future. And it allows for things like time series between the standard kind of quarterly sampling that fisheries folks tend to do. And of course, it's non-extractive and non-lethal and requires fewer permits to execute. So thank you for your time. I'd like to thank our funders and collaborators. Is there anyone out there looking for a job as an eDNA senior scientist? We're hiring, and I'll end there and turn it over to Professor Mouillot.
Thank you, Jason.
Can you hear me? Can I share my screen?
Yes, please share your screen, David, and we can hear you.
Is it okay in the present? That's okay?
Yep.
So thank you. I apologize in advance for my terrible French accent. So I am professor at the University of Montpellier in a lab working on marine biodiversity and exploitation. And I also hold a share in ocean about saving the unseen at the foundation of Montpellier University. And one part of my job is to monitor biodiversity of marine environments, including wind farms, using both eDNA and artificial intelligence tools. So I think there is a kind of consensus. This is the next generation of biodiversity monitoring with eDNA, both from the US, as you said, and the French National Biodiversity Strategy 2030, where we are supposed to monitor all freshwater and marine waters using eDNA. And when we look at all fields, it seems that eDNA has been used since more than 20 years, quite easily, but it was mainly on terrestrial systems like soil or lakes or rivers. In the marine realm, I must say it's much more recent, let's say 2015, 2016. I think because the challenge now is to filter a huge water volume in sterile conditions. So it's not easy to to do this task in marine waters and more complicated habitats than it was on terrestrial habitats. So now we try to get high standards to get the best result as we can with eDNA. So to me, we try to filter standardized 30 liters of water each time per sample. We have good solutions to fix eDNA a long time without freezing it. We extract eDNA in very strict conditions, sterile conditions, to avoid any contamination. We choose genetic markers which are well appropriate to your taxonomic groups, like fish, vertebrates, or mammals, or many others. And we try to get many sequences. We try to go up to 1 million sequences per marker to try to catch and detect rare samples, rare species. So we need most exhaustive as we can genetic reference database to be able to provide species list once you get your bioinformatic analysis. All this is done in collaboration with our Spagian private partner. So we have some pros and cons. I think we most agree about that. Pros are very easy. We can detect rare, elusive, and cryptic species, basically the whole tree of life, from virus to mammals. Non-invasive and safe, we are okay, very standardized, with a relatively low cost and sampling time. No taxonomic specialist, which is good sometimes, and can be used to monitor usually inaccessible habitats like deep sea. And we can use it as well as a time capsule to go back to the previous filters. For instance, we started in 2018 in the Mediterranean Sea. The genetic reference database was not as good as now, but we can come back to those old filters. So it means that if you start a program now, you always can go back to old eDNA filters doing more analysis, which is very valuable. What are the bad side? Weak correlation with population size. I agree with the previous presentation. We can have some correlation, but more like an idea of abundance or classes, but not exact match with population sizes. We have no information about life stage, size, or sex. Some species are hardly differentiated, whatever your marker we use. And you need a reliable reference database to make good assignations. So I think eDNA can supplement or complement other monitoring methods. We agree about that, like visual surveys, cameras, or acoustic. The advantage of eDNA is that we can use different filtering methods depending on the difficulty to access. It can be very easy on terrestrial systems with bags or very easy stuff, to more sophisticated tools to go to the sea or the deep sea, where we need large volume in sterile conditions. And wind farms are very challenging to sample because they are intricate habitat with deep elements, mainly for the floating ones we are trying to work with now. It's not easy with these cables and to go to 100 and 200 meters deep. We need new tools. So it's why we developed these new micro AUVs, which are small. I mean, small, 1 meter, 7 kilos, affordable, operational, and complementary. Because we try to get them working in groups. One is doing eDNA, another one is doing CTD, so physical-chemical parameters. Another one is doing a sonar to get abundance. Another one is doing a video. So the goal is to make the small AUVs to work together on the same site to provide complementary information. So I take the time to show you the last deployment we did some months ago in Corsica, Mediterranean Sea. It's not a wind farm because we didn't want at that time to try to investigate complex habitat. So it's a very easy site. So you can see the AUVs, quite small, very easy to deploy. And each one is doing a different jobs. Here, this is the eDNA one with a small filter you can see at the top. Very easy to drop into the water. You design your survey before on computer, and then the AUV is doing the job, going down if you wish. remaining in the surface, and doing beginning the eDNA filtering whenever you want, and then coming back to the boat very easily, very precise geolocalization. And then you will see in the nose of the AUV, the filter, which is encapsulated to be very strict with sterile conditions. So you get these kits in sterile conditions, you deploy, and you are sure to get water from the deep or for the site you are supposed to sample. And it can go on long distance, so you can drop it from the shore as well, and it can go back by itself to the shore. Here, it's a very small port where the AUV is finishing the trip. So we are trying to develop that more and more in 2025 in new places. So we have more expertise in seaports as artificial habitats. And our strategy is always to benchmark the biodiversity of these artificial habitats with marine reserves or nearby fished habitats. So it's good to have a comparison with the outside of wind farms parks. So here I'll show you some results on fish species richness. So you can see the purple one is a reserve, the green one is a fished area, and the yellow is a port. And we were doing the lockdown due to the COVID and outside the period of the lockdown. So you can see you have more species during lockdown in both reserve and fished areas. But when it's outside the lockdown, you can see that we have many, many species detected in seaports during this period. Very surprising that the biodiversity is the highest in port than in the outside. And when you look at commercial species, it's even higher. So it means that these seaports can act as nursery for commercial species, owing to their impossibility to fish and complex habitat structure. So you can host many juveniles, which can benefit to commercial fisheries when they get bigger. So the goal was to deploy the same strategy for the largest wind farm parks in France, Saint-Nazaire. It's a French big one, but not at your scale, perhaps. At the European scale, is not a big one, but we have 80 turbines, capacity of close to 500 megawatts, and it's 12 kilometers of the coast. So it's a big one at my scale. And we perform 18 eDNA samples twice a year, one in winter, one in summer. So you can see the wind farm parks in green here. We have some in blue samples inside protected areas. We try to keep the habitat to be the same. And we have some fished areas in green, in red here, to have a fair comparison. So we do the same number of samples inside the wind farm park, outside in fished areas and inside protected areas, to detect fish, teleost species, and chondrichthyans, so sharks and rays as well. But also crustacean species are targeted with another marker in these places. We do 2-kilometer transect with 30 liters of water filtered. So what we get with a very simple multivariate analysis is that we have totally different worlds. I mean, fish composition are very different between the wind farm sites in green, the fished zone, and the marine reserve on the left. So we have totally different species. So it means wind farms are creating a very particular habitat, which is not present in both fished and marine reserve, with many cryptobenthic species like the scorpion fish. While in marine reserve, you have more commercial fish like the solea, which is very expensive. And in these fished areas, you have more like mudgill species or mullet species, which are more keen to persist in human-dominated conditions. When we look at the species richness for fish, you can see that wind farms are between marine reserves, no take, and fished areas with a kind of in-between level of biodiversity. When we look at elasmobranch species, so very targeted species, very fragile, vulnerable, like sharks and rays, we are not so bad. We have even more elasmobranch species inside wind farm than outside, even within marine reserve. And we have almost no one in fished areas, which is expected. When we look at protected species like red list species, we see a similar levels inside wind farms than outside in marine reserve, and much more than in fished areas, which is expected. And when we look at commercial species, the same thing as for total species richness, we are in between, with more species than in fished areas within wind farms. So the next step for me is to take advantage of a new artificial intelligence method to take advantage of all this data we are getting now in a kind of a continuous flow of information. We try to assess the displacement or spatial dynamics of maritime activities when some wind farms are installed. We try as well to make some fish counts and identification of fish species on videos, underwater videos we can place in wind farms. We try as well to do some modeling, like counterfactual scenarios, to assess whether wind farms are changing biodiversity. So what can happen if wind farms are not there? And we try to as well model future scenarios under climate change to see what kind of biodiversity patterns we can expect under new conditions. So all these analyses are performed with two private companies, one working with eDNA, Spagen, a leader in France on that, and one Cyber, which is developing new tools like micro-IVs to deploy complementary tools to investigate intricate and quite deep environmental waters. Thank you for your attention, and I'm ready for questions.
Thank you, David. And thanks to both of you for these wonderful presentations. We've got about 20 minutes for Q&A, and we've got several in the chat, so I'm just going to start from the top. Let's see, where'd it go? And David, you responded to this question earlier, but I'll read it again, and then there was a follow-up to it. How far apart can the sample locations be while still being confident you are capturing the whole picture of biodiversity? David, you responded that.
Yeah, it's a critical question, because when I started 10 years ago, I thought that with sea currents and mixing waters, we could get a signal from far away, because eDNA is supposed to survive perhaps up to 12 or 24 hours. But in reality, maybe Jason has another idea, in reality, we lose the signal very fast. Some Japanese did some trials experiments. They have fish in a cage, and they know that when they remove the fish from the cage, they lose the signal after 15 meters apart. So it's very fast. And for us, we get a different signals from stations which are 1 or 2 kilometers apart. So it means that the signal is much more local than previously thought. Maybe Jason has another idea or experience.
Yeah, we've seen similar things. If you look at the half-life of eDNA estimated a day and the currents of a given area, you can calculate that you could potentially detect eDNA in the marine environment 10, if not tens of kilometers away from the source. But in reality, what we've seen is that, and we have seen Mommychug, for instance, which is a coastal species 20 kilometers offshore, but it was a trace detection. And I think that's what happens, is that those distal detections turn up as trace detections, because the DNA at the spot that was freshly at the spot you're sampling is going to dominate the sample. And you might get some trace detections from distal areas. It is a critical question, though, for interpreting. And it's always great to remind people that fish genes aren't fish. There's a difference in the way you have to think about sampling fish genes versus sampling fish.
And so as a follow-up, given that you're detecting the samples maybe in a more localized area, does that mean that your transects have to be close together in order to capture biodiversity, or what are maybe some examples of the distances between transects or sampling locations?
We tend to perform parallel transects inside the wind farms. So I mean, if a wind farm is 2 kilometers long, we try to make 2-kilometer transects in parallel. Each AUV is doing a kind of one transect, and then we go to, let's say, 200 or 300 meters. We do another parallel, and so on and so on. So we try to be quite exhaustive on the wind farm, making 2K transects and making them parallel, like replicates.
Our offshore surveys, we space the eDNA about 5 kilometers apart from each other, and I would think about redesigning that. It works, but I think we kind of overestimated or underestimated the spatial resolution of eDNA. The artificial reef data we've been getting back in other seasons shows similar ability to resolve the difference of fish over a 1 mile area of an artificial reef compared to the surrounding area. So that's kind of been like our field test of spatial resolution, one of them.
Is there a difference in reliability throughout the water column, say from sampling at the surface or a benthic habitat?
I can, so in our first study in papers that Mark Stokel led, using ocean trawl data, we sampled mostly at the bottom, and we did a comparison of surface samples taken in parallel, paired samples with the bottom samples, eDNA. And in 30 meters of water or less, there's very little difference in the eDNA results between the top and the bottom. We've continued to do that, and there is some difference. I've seen other presentations, other folks who have done surface bottom sampling over depth gradients, and showed that at about 30 meters, there begins to be divergence between what you find at the surface and the bottom of a water column. So in shallow water, it seems like there's not that much difference, surface versus bottom, in just simple biodiversity, alpha diversity indices. But it's going to depend on the environment, I would say.
Are there ocean conditions that affect the half-life for sampling?
Oh, yes. I think so. Even the solar energy, like UVs, like waves, like many things. So I think it's a very open topic to know lifetime of eDNA in seawater. But to me as well, I think it's lower than we think, except for some very rare traces.
Yeah, I'll add to that. Folks from, I believe, Ryan Kelly's group on the west coast of the U.S. presented eDNA degradation, and the stat I recall is something like 90% of the degradation was due to dispersal, physical dispersal, and about 5 or 10% due to biological degradation. Folks at the NOAA lab here on Sandy Hook have studied temperature as something that can increase or shorten the half-life of eDNA. And we did a study with Rutgers University looking at past surface current data, and they found that events can drive very, very widespread large dispersal events like hurricanes that we get in the fall here, just move water on the shelf so much that it really is a measurable impact on how much dispersal you might expect to be occurring in the eDNA signal on the shelf. So yeah, I think the ocean conditions matter from place to place and from time to time are important to understand if you're doing this kind of stuff.
For diadromous fish, can eDNA samples be assigned to a specific population if there's a reference sample that exists for an individual river population?
I'll David, you might have something else to say about this. I'll say in the way we're using metabarcoding, the markers aren't specific enough to identify different populations within the same species. I think I understand that question.
Yes. To complement what you say, Jason, what we try to do for instance, very rare species like angel sharks, we use metabarcoding to detect the place where it can be. And then we try to do afterwards some qPCR or over intraspecific variability with other markers to work on some specific populations. So the good thing with metabarcoding, you have a large screen. And then if you want to focus on some site or some species, still have enough DNA to go back to these filters and study more precisely some species. This is a great advantage of eDNA, that we can use several parts of the DNA filtered to carry out many studies. But we always begin with metabarcoding.
How does the cost of eDNA sampling compare to more traditional fishery sampling, such as trawls?
In terms of money or carbon?
Tough question.
Good question.
I assumed it was money, but if you have information on other types of costs, that would be great.
Well, David gave some information about the cost in euros for a sample.
Yeah, 600.
Yeah, and we do some of that in the lab and some of that in our lab and some of that in a for service lab. And the sequencing costs are about $100 per library per sample. But I think that the big money saving compared to capture surveys is the people time and the boat time. Ships capable of pulling a 30-foot trawl are super expensive to run on a daily basis, including the people required, and then the carbon footprint of that, not to mention, like David mentioned. So eDNA has the potential to be much cheaper than capture surveys. However, there are things in the capture surveys that cannot be replicated by eDNA analyses. I think that the challenge moving forward is to figure out how do we combine what we can do with eDNA, metabarcoding, and qPCR analyses to lessen the impact of and cost of fisheries monitoring plans and surveys, and make them better. For instance, add more eDNA and do less trawling, less days trawling, and focus the trawling on just the things that you really need trawling for.
And to me, in 2024, we should perhaps stop killing to survey or monitor. I think it's still shocking for us to, at the European scale at least, we try to ban all these scientific surveys now. So it's very strange to kill animals, to count them or to monitor them or to protect them. It can be useful sometimes, but I think with cameras or working with fishermen is better, but scientific surveys by trawling, it's not so useful, I think.
And one of the things that we're figuring out over here as wind farm development is ramping up and they're asking for monitoring plans and folks are calculating the sample size of trawling required to achieve the power necessary to differentiate inside versus outside. It's a huge number of trawls that need to be done, considering how variable trawl sample. If you're familiar with statistical power analysis, it can be hundreds of trawls per year necessary for quarterly sampling for one wind site. And that's a lot.
Has the citizen science project begun? And if so, how has it been successful?
It has begun. It's been successful in that we've been able to recruit and train people to do the sampling and participate. The data we're getting back does not look like our data sampled at nearly the same time and place. It's fewer taxa, lower diversity. There's the contamination I saw in the question. I wouldn't say contamination is the problem. I think it's the problem with we developed a different protocol for our community scientists to use that didn't involve freezing. We use self-preserving filters that are on the market. And when we saw these differences in their data compared to ours coming back, we did some in-house side-by-side comparisons, and it turns out that I can't speak conclusively about this yet. One half-student project has covered it, and another student project is ongoing right now, but it seems that we don't get the same amount of DNA off of those filters for the same volume filtered, and we don't seem to get, as you guess, in the metabarcoding, the same number of taxa off those filters. So we're rethinking our community science protocol to move forward. But that's minor compared to getting people interested in actually going out and participating in the sampling, which we have been successful in doing.
How many samples can the AUV take? And does it have the capacity to add preservation buffer to the filters?
Yeah, I tried to reply, but the AUV is performing only filtering one sample, but we try to launch two of them to have a replicate at the same time. Otherwise, we can add the buffer as soon as it gets back to avoid any contamination. So one sample at a time, and the buffer is just after the filtering, well placed in the filter.
This one was specifically for you, David. Do you have samples in the wind farm area prior to construction to be sure the difference in composition is a result of the development?
Yeah. This is the big challenge now. We try to go on every site where wind farms are supposed to establish in the next decade. So we try to have a huge mapping on the French EEZ to be sure we had samples before the establishment. So we are performing many surveys now on the French coast to get a before and after design.
Okay. What are the typical extraction? Oops, I lost. What are the typical extraction fixing efficiencies of eDNA?
Is there a question about the DNA yield? It's low. Tends to be low.
Yeah, maybe person can expand on that. I just read that directly from the chat. Let's see. Yes. So meaning the yield of the extraction kits.
From a leader, I'm trying to remember the units correctly. We get on the low end 10, the high end 100 or 200 nanograms per microliter. I think that's the unit from we use a Kyogen extraction kit. It depends where the sample's from. They're definitely low compared to like fin clip extracts or any kind of tissue extract of DNA. Then you have to consider that because it's an ocean sample filtered on a 0.45 micron filter, most of that DNA is going to be bacteria, and then phytoplankton, and then microzooplankton, everything but the DNA we're after. That's where the PCR amplification comes in.
Well, thank you both for taking on these questions. We really appreciate it, and for your time to present today. Before we adjourn, I just want to mention that we do have 2 other webinars coming up for WREN. One is on wind energy in Latin America. In Latin America, environmental solutions and best practices. This will be held in both Spanish, and we plan to have translation for Portuguese. That is on the 23rd of October. And then on November 6th, we have a webinar related to wind turbine design and bat interactions. Thank you again to David and Jason. Thank you all for participating today. We hope this webinar was informative for you. Certainly was for me.
Thank you, Chris. Thanks,
Have a nice day. Bye-bye.
Bye, everyone.
Bye, Jason. Good luck.
Bye.
Cheers.
Cheers to you.
Yes.